my screen?
That's all okay.
Good. Okie doke, let's get started. So back in October last year, Marco already gave an introduction into knowledge graphs and graph databases. So my talk is following on Marco's talk, and is going to talk specifically about the knowledge graphs we're building for KnetMiner and present some use cases. We recently published KnetMiner in Plant Biotechnology, so if you want to read more about it, that's the reference. Okay, so all right, let's start with the challenge that we are actually trying to address with KnetMiner. So, as you know, many of us at Rothamsted, many scientists at Rothamsted are trying to understand complex traits and diseases in a variety of crops, from wheat to rice. So to do that, they design experiments to try to narrow down the genes and gene networks that are often controlling many of these complex traits. So the experiments they design fall into two categories: genetics, for example, QTL studies or genome-wide association studies, and the other range of experiments are, I call it omics studies, so covering genomics, transcriptomics, proteomics, metabolomics. So genetics and omics studies really help us to narrow down from the variety of genes that we've got in a genome to a much smaller search space to say, you know, this likely to be candidates and behind a certain complex trait. So this step, I call it data generation and event detection, which we have now more or less mastered. Oh, well, it's still a very challenging task, obviously, to do the analysis, but it's getting less of a bottleneck. Where the main bottleneck sits, in my opinion, right now, is to go from this step to the next step, which is about the interpretation of the data that comes from genetic and omics studies. So what do I mean with interpretation? Interpretation means putting your own research into context with other published work. So it could be that, for example, another researcher has done a very similar study in wheat and has published the data already. So it would be good for us to be able to relate our data to another study in the same crop. But often, you know, not exactly similar studies have been done in my crop of interest, but maybe somebody has looked into a similar analysis in a very different crop. So being able to get a good overview of all the different types of research that have happened that could help me to interpret, prioritize, and generate reports more efficiently from the data I generate and from the analysis I perform. That's, I think, where the bottleneck is right now. So really what we want to do is to be able to organize the knowledge so that it is possible to go more smoothly from data generation, event detection, to the interpretation and idea generation. So with KnetMiner and my team, we are trying to organize the knowledge about biology. What sort of knowledge are we looking at? For example, gene SNP trait relationships, gene QTL trait relationships, protein-protein interaction data, information about genes and their function, information about what happens if I knock out a gene or overexpress a gene. So relationships between genes and phenotypes are very important type of information that we like to organize and bring into a structured format. Gene publications, protein pathway information, gene regulation information. So a transcription factor is regulating the expression of a certain gene. gene expression or co-expression information about proteins and how they interact with compounds and small molecules. So all this is very important information to try to organize and model, and obviously information about other species as well. So not just looking into wheat, but also considering information about model species or related crop species. So to do this, we use the concept of a knowledge graph to organize information and knowledge. So what do we mean with a knowledge graph? Let me just give you a quick introduction to knowledge graphs again, and this is really a recap from Marco's talk. So a knowledge graph consists of nodes and edges. So in this example, I've got 3 nodes, Rothamsted Research, Harpenden, and Norswick. Each of these nodes has a label. So in this case, we've got a label Research Institute, and Harpenden and Norswick are sites. And nodes are connected through edges. Each edge will again have a label. So in this case, the relationship is called based in, and the relationship between Harpenden and Norswick is called what we call it distance to in this case. The last bit about knowledge graphs is that every node and edge can have a property. So we've got 2 properties in this model. So we've got a year property and we've got a miles property on these specific relationships. So this is what we call a knowledge graph or a labeled property graph. And there are lots of ways of modeling knowledge. So for example, in this case, I added a property year to my node Rothamsted Research. I could have modeled it in a slightly different way. I could have said, instead of using a property, I actually go and create a new node called 1843 of type year, and a relationship that Rothamsted Research was founded in 1843. What that allows me to do is that I can now come and connect lots of other things that happened in 1843 to this node. So this is knowledge engineering, and often requires an experienced knowledge engineer to help you make decisions and design choices about how to model knowledge in a most efficient way based on the applications you're trying to build. So there are many types of knowledge graphs out there, and I'm just giving you a few examples. The most famous one is Google's knowledge graph, which is about lots of things that are happening in the web. Then there is, for example, a research graph, a research knowledge graph, which has 4 main entities, and this is how the schema looks like. At the center, there is a node of type research data, and this is connected to nodes of type researcher or nodes of type publication and nodes of type grants, and they have relationships between them with different properties and so on. So this is a helpful knowledge graph to help us share knowledge, share publications more efficiently, and enable better collaboration between researchers. Then there is a knowledge graph that NASA, for example, is building. It's called NASA's knowledge graph, which is more like a lessons learned knowledge graph. So they've been building since the first experiments. They've been modeling all the lessons they've been learning into a knowledge graph to improve future predictions. So it's just another type of a knowledge graph. So the knowledge graphs we are building with KnetMiner, they are all about trying to organize biological knowledge. So I've shown you the types of information we are interested in. What we do next is to try to identify data sources that actually represent that sort of information, like, for example, gene regulation information or gene expression, gene function. So these are some data sources that are useful for plant biology and for building the wheat knowledge graph. So we take this data, we use the tool NetBuilder to transform it into a knowledge graph, and this is how the knowledge graph looks like. We've got nodes of type gene, for example. that are differentially expressed in a certain study, and you've got nodes of types phenotypes and traits and so on. So I'll go into this in a second. In total, we bring together over 40 data sets for 110,000 genes. The size of the knowledge graph that we've built so far has about 750,000 nodes, 4 million edges. It's open access, available in RDF, Neo4j, and some limited REST APIs out there as well to query the data. Okay, so let me just do. Let's see if I can switch. Can you still see my screen, or did I just share? So I'm trying to show now my browser. Yeah, you see my browser, Andrew? Okay.
Yes.
Okay, so let me just show you an interactive version of a knowledge graph. Which one do we take? Let's take this one here. So the knowledge graphs remember again, so we've got different types of nodes and edges. So at the bottom here, for this specific knowledge graph, you see the different types of nodes we see here. They represent SNPs, genes, traits, proteins, publications, SNP effects, protein domains, pathways, co-expression clusters, plant ontology terms, co-expression studies, biological processes, functions, enzymes, pathways, reactions, and so on. So large range of different types of information have been put together into this example. So now let's zoom in and just show you what, for example, happens if we click on a certain node in this knowledge graph. So I'm going to select, for example, this node here. You see this nodes here I'm selecting, they are of type publication, and they contain lots of different properties. For example, a source property, evidence property, a year when this paper was published, the title, the abstract, and so on. Let me click on a different node, for example, this node here, or hang on, just go on the wheat one actually. So on this one, so this is the node that's representing a gene, and it contains again different types of properties like a tax ID, start, stop, chromosome information. Okay, and then finally, let's click on a relationship. For example, let's zoom in and pick this one here. So this blue relationship is of type co-occurs with. What that means is that this node and this node, they co-occur together in a series of publications. These are the PubMed IDs. And the co-occurrence relationship has a strength or has a score. So the score can be again added as a property to the relationship. Okay, so that gives you just an idea of the content of NetMiner knowledge graph. I'm switching back to slides. Back here. All right, so why is it of advantage to use graphs in general to model data? Well, graphs are a really good data structure to model complex, interconnected, semi-structured information. That's so biology is quite noisy and complex, so they're really well suited for modeling biology. Then the other advantage is that once the data is in a graph format, then you get access to a range of different algorithms to search and query the data. For example, path-finding algorithms to find the shortest path between two nodes, or centrality measures to determine which of the nodes in the knowledge graph or in the graph are of higher importance because they've got more incoming or outgoing edges, or community detection based on clustering your graph into parts. So graph algorithms have been around for nearly 300 years now. In 1736, Euler laid the foundation of graph theory, but it took hundreds of years for the first graph algorithm for the graph theory book to be actually published. Graphs are still one of the trending topics. So recently, in one of the reviews about data analytics, graphs were seen as one of the trends in 2021, although they've been out for so many years. So what I'm going to do now is to show you some case studies of how the knowledge graphs that we are building are exploited with NetMiner to actually drive biological questions. So the first case study I'm going to focus on is the forward genetics approach in crops. I think I've picked tomato and one in wheat and Arabidopsis. Okay, so I don't know how familiar you are with the term QTL, so just a brief introduction what a QTL is. There are different routes to gene discovery. As I mentioned in my first introduction slide, one of the things that scientists do is trying to identify regions in the genome that can correlate with a certain phenotype. These regions are called QTLs. So they're designed, they start with the mapping population, they cross two different parents, they show different types of phenotypes. For example, in this example, we've got leaves with lots of hair and leaves with few hairs. And then in the progeny, they can observe different phenotypes, a lot of hair or very little hair, as you see in these examples. And if you look carefully, you can see that whenever we observe very little hair, we have this blue block on the chromosome. So here in this example again, here little hair correlates with this blue block, little hair correlates with this. So using this correlation between the genotype and phenotype, we can find regions that are called QTLs. So the problem is that these QTL regions can contain hundreds of genes, and it is very challenging to first identify what the functions of all those genes are and find an unbiased and systematic view to prioritize those genes to see which of them actually is the causing gene for my trait of interest. Or sometimes it's a complex trait and it's not just one gene, it could be a gene network. So how would you go about reviewing the literature for 100 genes? Which resources and databases would you go and use? How long would it take you? We estimate, for hundreds of genes, if you're not expert in the domain and the range of databases that are out there, if you wanted to consider them, it would be, it can take a PhD student easily a month or more to review 100 or more genes. So I'm going to show you an example how this can be automated with NetMiner. So the trait I'm going to take is potassium content in tomato. The NetMiner user interface allows me to define a QTL region. So for example, here, my QTL is on chromosome 12. It starts at this base pair, ends at this base pair, and it contains about 115 genes in that region. So I specify my QTL. I explain what this trait is about. So it's about potassium content. So I use keywords to express my knowledge about this trait. I press the search button, and what happens is NetMiner evaluates all the knowledge about this 115 genes that are in the knowledge graph and uses a gene rank algorithm to rank them. This is the top ranking gene. It's called POT5. It's on chromosome 12 within this region, and the evidence that NetMiner presents why POT5 is highly ranked is what you see in this knowledge graphs. So POT5 is the tomato gene. It has an ortholog in Arabidopsis, and the Arabidopsis ortholog has a strong relationship with potassium content, which is coming from a certain paper that was published recently. Okay, just a warm-up. This is a warm-up example. So again, I'm going to switch now back to live to show you a second example, this time in wheat. Okay, so this is the NetMiner interface for wheat. I'm going to use one of these examples we've got on the right-hand side. If I click on it, it populates the search interface with my search query. So in this case, I'm going to analyze a QTL about grain fill duration, and I'm going to use the keywords grain fill duration or seed growth, and I'm going to define, I have identified in my QTL analysis two strong QTLs, one on chromosome 5A and one on chromosome 2D. One has 123 genes, the other one has 109 genes. And quite often with these complex traits, we do get multiple QTLs, and it would be nice to see if there are interactions that are happening on molecular level between those QTLs. So I'm going to search the knowledge graph now with my query. So all these genes have been now searched in NetMiner. The knowledge about those genes has been evaluated, and I'm getting a list of ranked genes presented in GeneView. So what I like to do now is I want to take the top ranked gene on chromosome 5A on my first QTL. It's this one here. I select this one, and the top ranked gene on chromosome 2D on my second QTL is BPM5. So I'm selecting these two genes and asking NetMiner now to generate me the network for those genes, so I can see if there are any connections between those two genes. Okay, so here we are. So NetMiner has done two things here. It has first identified those two genes in the knowledge graph, has performed lots of different graph queries to generate the knowledge graph for these two genes, ADA2B and BPM5. But then it has done another thing. It's done some filtering. So the knowledge graph for those two genes has about 2,000 nodes and 2,500 edges. But because we do not want to overwhelm people with all this information, it has hidden lots of information and has just kept the most important things on the screen. So what I'm going to do now is run through the information that NetMiner presents and start adding other things to the network. So the most important bit of information I see in this graph is that the two genes that are located on two different chromosomes, on two different QTLs as well, they have a relationship between them. So this relationship seems to be, let's check, it's a regulates relationship. So there seems to be some regulatory role between these two genes. So one is a transcription factor and one is its target. There is a weight associated with this relationship because it's a predicted relationship. It's, it could be true, it could be a false positive as well. So we need to be aware of this. Now let's study the evidence. Why was this gene linked to grain fill duration? So we see there is a link on this side here. It's based on, if I click on this, you'll see this node here represents a trait ontology term based to maturity or grain fill duration. And the evidence that links this gene to this trait ontology term is coming from a GWAS study. So the story is that this gene has certain alleles or certain variation. So this is a variation. It happens at this base pair level. And this SNP is associated with this trait based on a GWAS study. And the p-value of the association is shown here. So this is why my first gene was related to the trait I was interested in, and then the second gene I had selected is also related to regulation of seed growth, but the story here is a bit more distant, and indirect. So you have a link that goes from the wheat gene to Let's add labels to it. It goes from wheat to Arabidopsis, a model species. The Arabidopsis gene is interacting with another gene. This is protein-protein interaction data. And the interacting partner is actually related to regulation of seed growth. Okay, so what I can do now is I can study this network in a lot more detail. I can hide information to it or delete things from it. So to hide more information to it, I can ask the question, for example, in which co-expression cluster are these two genes? So I can use this legend and I can add things interactively to the network, and you'll see new nodes appear on here. I can add more traits to it to see what other traits this SNP, for example, has been associated with. And I can delete things from the knowledge graph as well by doing similar functions, double-clicking on things here or right-clicking and hiding information. In the end, I have explored the knowledge and I like to save and share it with someone. So I press this Save Network button here, which uploads the network into my NetSpace. And this was where I started originally. So this is my NetSpace. It has a collection of things I have worked on. And this particular network I've saved, I've called it Monogram 2021 as a demo for the conference. So this is the network, the knowledge graph for these two genes, the analysis I've done, the things I found as important and I want to flag up and show to a collaborator. They are shown here now. NetSpace allows me to share this with a public URL to someone. So I could take this link, hang on, and paste it into our chat here. Let's see if that works. Okay, here it is. So DSRG, this is a really interesting finding I made. Please go and check and see what you think about it. I'm not going to tell you in a long email what all these interesting relationships are. I'm going to share the knowledge graph with you. It has all the information. So click on that link, go on this button here and start exploring the network yourself to see if the relationships that are shown make sense to you as well. So switch back. I've got too many tabs open here. It should be this one here. Okay, so what did we do? So we started with 2 QTLs. We narrowed it down to 2 genes that were top ranked in those 2 QTLs. We could see there is a gene network. And the different types of information I looked at, like circled in this example here, they were all coming from different data sets. So we had information coming from ensemble plants, we had information coming from biogrid, we have information coming from gene ontology annotations. This top information is coming from a database called T3. And there were some relationships in the knowledge graph that are not present at all in databases. They were generated by NetMiner. So we augment the knowledge graph using text mining and add new things to it ourselves as well. It would have been very hard for a scientist who is not expert, and even for a bioinformatician who is an expert, to do this sort of analysis and integrative analysis. So NetMiner saves a lot of time. Okay, so moving on. How am I doing with time? I've got a few more use cases, so I run a bit quicker through this one, just to give you an idea of how certain types of data are integrated into the knowledge graph and how they are visualized. So I'm going to talk a little bit about genome-wide association studies. It's similar to QTL studies, just using different populations to identify regions in the genome that are associated with a trait. This GWAS studies can sometimes be a bit noisy, and identifying genes in these studies is very challenging as well. So how do we model the data in our knowledge graph? So GWAS data looks a little bit like this. You've got different types of information. You've got SNPs, so polymorphisms in the genome that are associated with a phenotype. The phenotype is often free text, unstructured, described like this in this column here. And we've got a p-value for the association. We've got some additional information like which genes are in close proximity to this polymorphism and how far away are they from this polymorphism. So this specific data set I've taken has about 66,000 SNPs, 20,000 genes, and 107 phenotypes. Okay, so what we do is we use our toolkit, NetBuilder toolkit, to transform the tabular data into a graph representation. We model the data like in this example like this. We generate three types of concepts. One is a gene, one is a polymorphism or SNP, and one is a phenotype. The relationships between them, we call them a gene is close to or in proximity of a SNP, and the SNP is associated with a trait. So, and each of these relationships can have properties like p-values or a distance. So if we carry on bringing in more data into the graph, we can now start seeing connections. Like in this case, two genes are linked to the same trait. We can now overlay other types of data on top of the GWAS data. In this case, this is protein-protein interaction data. If we overlay it into the networks, it looks like this. We see new connections happening that can help us to understand the biology a bit better. I don't want to go too much into detail about it. I want to focus a bit more about, so how do we integrate GWAS data and phenotypic information? So there is a very good database called Arapheno, which contains a phenotype metadata like studies, DOIs, mappings to ontologies like trait ontology and environmental ontology. So all this sort of information is very useful to have in the knowledge graph to help us integrate data more efficiently. So once it is in the knowledge graph, NetMiner has different ways of visualizing GWAS data in to the end users. So there are three things I've highlighted. If you search for a keyword like anthocyanin, there is a suggester coming up here that it shows you anthocyanin is matching to several different GWAS studies. They're mentioned here. The other piece of visualization that NetMiner uses is when you get to your ranked list of genes and you can click on these icons here, it would tell you that some of these evidences are based on GWAS data and which GWAS study specifically it was. And the third one is there is a view called map view, which shows you the chromosomes and the genes on the right-hand side, and then on the left-hand side it visualizes the different GWAS studies that are in the knowledge graph as well, which is a nice and interactive way of seeing overlaps between predicted candidate genes and published GWAS data. Okay, so then once we dive into the networks, we can, for example, focus on a specific study called anthocyanin 22. This is one of the Arapheno studies. It looks like this on the website. Then in NetMiner, we can ask the question, so which SNPs are linked to this trait? Which genes are in proximity of those SNPs? So this is really powerful already, but then it goes even beyond that. So now we can ask which other genes or proteins are interacting with these initial genes. So it can show you that info layer as well. And if this is not enough, we can ask the question, so this is all Arabidopsis data, and what are the wheat orthologs? So it goes and adds you that layer on top of it as well. So there are 4 or 5 different data sets that have been integrated here and can be visualized in one place. Okay, So I've been talking about forward genetic studies, how to go from QTLs to genes and gene networks. And now I'd just like to show you a reverse genetics example. In this example, we've got a knowledge graph that we have built from lots of different public studies. We have integrated data in a smart way. So now we can ask the question, so what happens if we knock out a specific gene? If we designed an experiment where we knocked out a specific gene, for example, in this example, it's an TT2 gene or RMYB in wheat. Can the knowledge graph tell us what phenotypes we might be observing without actually doing the experiment? So let's open the knowledge graph and have a look what it's linked to. So it's showing us a few interesting things. So TT2 is this gene in the middle of the knowledge graph. We've got relationships that are showing us that there is some gene regulatory information relationship between this gene and a few neighboring genes. And the neighboring genes, they are linked to things like color, pericarp color or grain color, or seed dormancy and grain germination. At the bottom here, we can see the Arabidopsis ortholog of TT2 has some relationships to root hair length, root number, root hair density. So, okay, so 3 different types of traits: color, dormancy, germination, and some root-related traits. So actually, I picked this example because this gene is well studied and it has been knocked out, and the phenotype of this gene is well known. So what happens if you knock out this gene in real is that you observe 2 things. First is that the red grain color changes to white grain. So that's a positive trait because the white grain has more favorable taste for many consumers, especially children. But there is a negative trait that happens as well when you knock out TT2, is that you observe pre-harvest sprouting. So this is when the grain starts germinating too early. This is a negative trait and not great. The link between those 2 traits has not been identified yet. So people really did not understand on a genetic basis why these things are coupled, but they go always together in a pair. And with this example I was showing, we could see that maybe that's just because of an interaction between 2 genes. Beyond knocking out, we can design an RNA-seq experiment, and this has been done as well, to study which genes are differentially expressed in controls versus RMYB mutants. So an RNA-seq study that was performed in this case has generated lots of differentially expressed genes. What we did is we compared those genes with the genes that are in our knowledge graph in close proximity to TT2, as in this example, to see if there are overlaps. And we could see high overlaps between genes in the knowledge graph and the genes that were differentially expressed. So this is a reverse genetics approach showing you that there is a lot of information that can be derived and inferred from the knowledge graph without actually doing the experiments, potentially, if the knowledge graph is very rich, powerful, and has clean data. So to summarize, visualizing connections can lead to new lines of inquiries. In this example, grain color and pre-harvest sprouting might be linked because RMYB targets, because our genes and MFT might be interacting with each other. That's something that is not studied yet, but looking at the evidence and the networks, it might just give you ideas to think this way. The other question scientists might ask by looking at the knowledge graph is, do white grain varieties have increased root hair density? Again, something that nobody has thought about, looking into the underground phenotypes and traits as well for our mutants. So it would be interesting to study the number of root hairs for our mutants. And is there a link between root hair density and pre-harvest sprouting? Is maybe the pre-harvest sprouting happening because the root hair density is different in different varieties. So lots of interesting questions that someone can, ideas that NetMiner helps scientists to generate. Finally, a case study, I'm going to jump into COVID-19 and a very simple RNA-seq study. Do not quiz me about the design of this RNA-seq study. I can just tell you, so it's about a transcriptional response of human lung epithelial cells to SARS-CoV-2 infection. So they have compared infection versus mock. I have no idea what mock is in this case, but let's just, not think too hard about the design, but just take the number of differentially expressed genes that come out from this study. I think it was about 50 in this case. And the question scientists would then ask is, are any of these genes already known to be related to COVID-19 or to coronavirus infection? Because, there are every day so many COVID-19 related studies that get published. You really want an efficient way of relating your own experimental data to the wealth of new published data about COVID. So you take the list of differentially expressed genes, paste it into NetMiner, use NetMiner in an iterative way, first without keywords, to just search and see what sort of knowledge is overrepresented in the knowledge graph for these genes. So it shows you the few of the overrepresented terms that pop up in the knowledge graph with a p-value and how many of the genes from your list are linked to them. Then you can go and refine your search and add some of these keywords to the top bit here and ask NetMiner more specific questions about, show me linkages between my genes now to specific drugs like remdesivir or defense response to virus. And NetMiner shows you the evidence in these views and allows you to generate very quickly a network of some of your differentially expressed genes and how they link up to defense response to virus or to certain drugs that are published in certain papers and so on. Okay, so this is, so we've covered quite a lot already. We covered knowledge graphs. I've shown you some use cases. Now, just in the last few minutes, I'd like to say a few things about our roadmap. One of the things we really like to do is to start building multi-species knowledge graphs. So what I mean with that is right now our knowledge graph, for example, for wheat has wheat data and Arabidopsis data, but it would be really nice to bring in a variety of different crops together into one knowledge graph. So for example, if somebody's studying tomato, information about potato and pepper are equally important. So scaling the knowledge graphs up to multiple species, related species, is very important. The other area we like to explore a lot more in future is pan graphs. So modeling genetic differences within species. There are many different types of genetic variation within species. There are things like presence-absence variations, copy number variation, structural variations, SNPs, indels. How can we model that sort of information in a knowledge graph is one question. How can we model metadata about single plants and cultivars in the knowledge graph? So I think understanding how to model it properly is key, but then also building the pan graph analytics tools to help scientists to go from gene mining to actual allele mining, which is the next big challenge. So these are two important areas. And the third area we like to focus on is building much richer NetMiner APIs and microservices that allow you to use the data that we are generating and integrating in lots of different types of applications beyond just NetMiner. For example, it would be nice to build Galaxy workflows that have NetMiner as one of the steps. So this is a workflow that goes from, it's an RNA-seq workflow. And at this step, normally people would stop where you do your differential gene expression analysis, but we could feed that list into NetMiner and use a knowledge mining approach to rank the genes. We could connect to the knowledge graph through Jupyter notebooks, for example, and do, for data scientists, run your own queries and search and graph algorithms on top of the data. For software developers, this would allow them to build a lot more interactive, reactive, and nicer visualizations on top of the knowledge graph, powered by technologies like GraphQL. So to enable us to go really from what we have now to a NetMiner that is multi-species, pan-genomic, and has much richer APIs, we need to radically re-engineer NetMiner. We've been evaluating different ways of how we could do this, and one of the stacks that we are currently considering is called the Grand Stack, where we've got a dedicated graph database which will be holding all our data. I'm estimating the new knowledge graphs that we might be building in future to have about 100 million relationships that need to be indexed and stored efficiently in a graph database. Then powered by a middle tier called Apollo GraphQL, which serves the data efficiently, and then React front-end tools to visualize it. So yeah, just to finish up, this is the NetMiner team. Ajit, Marco, Jeremy helped with the core NetMiner development. Joe was part of the team, he's left. Dan is helping us to test tools and build applications and has really good ideas about new features that can help to drive use cases in DFW. Chris is our scientific advisor. And obviously, the fair data, we are consumers of fair data. So NetMiner would not be possible without lots of people putting hard work into creating fair, curated data that can be nicely integrated into these knowledge graphs, on top of which we can build useful applications like NetMiner. Okay, with that, I'll stop. And if you'd like to read more about NetMiner, check the publication.
Thanks, Keywan. Oh, an echo.